Hey Bioteam. So you guys know that ribosomes are responsible for building proteins using the mRNA that they receive from DNA. So in the case of this cell, the DNA would transcribe a strand of messenger RNA, and the ribosome, this little guy right here, would take that messenger RNA and use it to translate out a protein, a chain of amino acids, or a polypeptide. But in eukaryotic cells, there are two types of ribosomes. There are free ribosomes, which are called free ribosomes because they're floating in the cytoplasm. And these are responsible for translating proteins that get used inside of the cell. So we'll show an example of a protein here. But the other type of ribosome is a ribosome called an attached ribosome. And so you can see these attached ribosomes right here are attached in the rough endoplasmic reticulum. And in fact, the rough endoplasmic reticulum gets its name because the ribosomes give it a rough looking surface, kind of a sandpapery appearance. So these ribosomes are responsible for translating proteins that the endoplasmic reticulum will then transport to the Golgi complex, which is down here. And once the proteins make it to the Golgi complex will modify and fold those proteins and then package them inside of vesicles before shipping them off to one of several places. And vesicles are these tiny little membranes that you can see kind of folding off of the Golgi bodies, kind of pinching off of the Golgi bodies here. And the place that these vesicles get shipped to depends on the type of protein inside of them. So some types of proteins get secreted out of the cell entirely, and we call these secretory proteins. And you'll notice that as the protein gets secreted out of the cell, it loses its vesicle in the process. The vesicle actually ends up merging with the cell membrane. If the protein getting packaged happens to be a membrane protein, that protein then is wrapped in a vesicle and shipped over to the cell membrane, where it actually embeds itself into the cell membrane. And it acts as a channel or a receptor. So it either lets things through or it receives signals from other cells. And last, if the protein being processed happens to be a hydrolytic enzyme, or an enzyme capable of doing hydrolysis, of breaking things down, once those enzymes become wrapped up inside of a vesicle, they actually become a lysosome, because a lysosome is literally just a vesicle, a tiny little membrane, filled with these enzymes capable of doing hydrolysis, capable of breaking down and recycling other molecules within the cell. As for why our cell would wrap proteins inside of vesicles in the first place, it has to do with the fact that proteins aren't able to get through membranes by themselves. They're too big to fit through the phospholipid bilayer. But by wrapping these proteins inside of a vesicle, the vesicle can actually merge into other membranes and kind of push the proteins inside of them through that membrane. And the reason it can merge is because it is also a membrane itself. So it can kind of incorporate itself into the bigger membrane and then push whatever's inside of it through. And that's it. At this point, you guys have some practice problems. We'll see you next class.